go back to the days that I had you. Back to one more night like we used to. I miss every minute we've been through. From the hardest part to the best you. Make the stars align. Words cannot describe. Don't need no more goodbyes. You leave me hypnotized. Make the stars align. I need you here tonight. Don't need no more goodbyes.
So what is a species anyway? Here on our left, there's an eagle, or an African fish eagle, whatever. Okay, and on the right, yeah, it is more familiar. It's that the emblem of the United States of America, bald eagle. Yeah, I have seen several times this particular eagle, but I can't without this label. There's no way I can distinguish these two differences. Look, they all look the same to me, but obviously they are two different species. African fish eagle is independent species from the bald eagle. So, what's the criteria of like deciding whether this eagle is different species from the bald eagle? And what about this? These snails, it's all different. There are obviously we can recognize that there are different, but also what we know is they are an individual member of the same species of a snail. And in the case of these all different human faces, which all portray the uniqueness and differences, but we all know that these are all different individuals, but they are all in the same species of Homo sapiens human. So this is intra specific differences. So what distinguishes these different species versus differences present and then found in a single the same species? Here, there's another example. A dog. All dogs, all modern domesticated dogs, or actually derived from wolf, gray wolf, yeah, particular type of wolf. So, dog and gray wolf actually share a common ancestor. Okay. Still, actually, dogs and gray wolf are not considered to be independent, different species. They are still technically regarded as the same species. And so even though you can actually plot here on a single common ancestor form, but still they are the same species. And even though all these physically so distinct different forms of dog breeds, but still each different form of dogs are the members of the same species, the dog. Yeah, they are all, even with this gray wolf and all these different breeds of dogs, they share the same common scientific name of the Canis lupus, yeah, indicating that they are the members of the same species. So here is one version of the species definition. This particular version of the species definition is called the biological species concept. Biological species. It states that a group of organisms that can potentially interbreed. So interbreed is the keyword. They can mate each other. Interbreed with one another to produce, but this mating is not good enough. Once you can mate, then another thing is then should be able to produce a viable, healthy enough to live, and also fertile, that healthy, not only being healthy but also being able to produce their own offspring, so fertile offspring. And that is the condition, the minimal condition of any members belonging to a same species group. So that is a specific way of defining species we called the biological species definition. There are some other different ways of defining the species. However, it is the most widely used uh, definition, and also it is very intuitive, at least intuitive and convenient as far as like when we try to define the animals, like, and those who live their lives in based upon the sexual reproduction, and it is very straightforward to define all those sexually reproducing organisms. Okay, so what I mean by that, under such definition of a biological species definition, what I mean by that, those two members cannot uh, uh, interbreed. What prevents any two individual from being able to interbreed? So, uh, well, if they were kind of blocked, so this is called a reproductive isolation. So, if two individuals were somehow isolated in terms of their reproduction, whether it's an opportunity or kind of a physical or biochemical characteristics, if they were blocked, they will not have the equal opportunity or being able to perform this reproduction, then we said uh, they were reproductively isolated, these two. And this reproductive isolation is the starting point of generating two new independent species. So once again, we can say that reproductive isolation is a key determinant for generating the new species during the course of evolution, of course. Like once you, as we have seen in previous lecture, once you have formed a new species, then you accomplish a new small accomplishment of evolution. So such barrier, what prevents from mating? Like I said, there are different ways of preventing, like in the form of blocking the opportunity, or in the form of like making physically or biochemically impossible for. So those barriers, several different types of such reproductive isolation barriers exist. We can group two different categories of such reproductive isolation. One is called before, pre-zygotic. Zygote is what? The sperm and egg combines together to restore this individual, so that from which through the cell division, the new individual is formed. That is a zygote. We have seen this term in the first lecture of the semester, right? Zygote. And the gamete, another opposite term, a gamete, two different gamete, either like a sperm or an egg, but there are two different types of a gamete. Two gametes combined to form a zygote. That's how you do this sexual reproduction. So this pre-zygotic barrier means something is blocking so that they cannot successfully form this zygote. Then that is all considered to be one of such mechanism of pre-zygotic barriers. And on the other hand, post-zygotic barriers means even though these gametes were able to be formed, okay, so gamete was formed. I mean, the zygote was the gamete fusion to lead to the formation of the zygote was successful. But even afterwards, something is happening, so that development, successful development afterwards is prevented is what we consider as so-called post-zygotic barriers. Okay, so here the point is, important point is, you try to recognize each individual cases and then to attach them. Oh, this is the pre-zygotic and this is a post-zygotic. That is something a little less smart way of studying and understanding this. Understanding is the most important issue, and the way you soundly understand these all differences is once you have, there are two different ways of having blocking the reproduction. You could like intuitively think the ways. Well, you could like block those two gametes meet. Several different ways of you achieving this prevention of a gamete encounter. This will be an example of pre-zygotic barriers. And also you could think of the occasions where, yeah, okay, well, you have this gametes met, but then still you have some kind of a mechanism by which you interfere with the proper development of this offspring, then all those should be considered to be one of such post-zygotic barriers. As long as you can distinguish like logically, then why do you need to memorize all those different cases? Is something that I really want you to grasp and understand these things. So here, even though under the pre-zygotic barrier, 1, 2, 3, 4, 5 different mechanisms been introduced, and on the other hand is under the post-zygotic barrier, you have 3 different ways of achieving these post-zygotic barriers. But just understand and okay, this using in this way you could, there is a certain way of preventing those zygote formation, like just by understanding this, each of this mechanism will be just sufficient, so that you should be able to explain. Actually, one very good way of testing yourself whether you understood this or not is, at this point, using this figure, you try to explain each different occasions and why these occasions, like represented by these 2 flies, would be an example of so-called what? Habitat isolation. What is that? Is probably the way you check, self-check yourself, the level of understanding of this. So, like I said, there are 5 different categories of such pre-zygotic barriers, like including habitat, temporal, behavioral, mechanical, gametic. But just by looking at, hopefully, by looking at this wording, you can understand habitat isolation. What do you mean habitat isolation? So habitat isolation means the place of living was separated. Then obvious, though, if those 2 individuals were like isolated in their place of living, and then opportunity wise, they were not given any opportunity to have the zygote formation. So if that is the case, they will never be able to produce. So they will never be able to fulfill so-called this biological species definition. So they are not a member of the same species, obviously. Temporal means timing wise. Yeah, those 2 individuals, even if they live together in a nearby locations, but if their activity of this again attempt of sexual reproduction are somehow different, time-wise different, but they will never be able to give any opportunity. Behavior is also certain organism will only accept a certain specific behavior in their mating attempt, and that would be like understandably so. So if their behaviors of like maiden ritual, courtship, we call that as a courtship ritual. If the courtship ritual is different, then they will never be able to have this successful gamete production. And mechanical isolation, you can possibly understand this, like this event of actual fusion, gamete, sperm and egg fusion, should be done through some specific their organ, sexual organ. But if their sexual organs are not compatible, mechanically speaking, then they will never be able to do this. And gametic isolation is a kind of the chemical, biochemical barrier. Even if mechanical they were able to do this, but if sperm and egg of these two different individuals are not compatible so that they have this fusion, then it's finished, is the thing. So this habitat and temporal and behavioral isolation, all these three are, or hopefully you can understand, all these three different barriers belong to the prevention of this so-called the opportunity is blocked. Okay, they don't even have any chance to have this mating attempt is blocked. So this habitat isolation, I will just go a little in detailed explanation, which is actually unnecessary, but I will just do it anyway. So another name of calling this habitat isolation is ecological isolation. So two closely related species, although they will, but even if they are very close, but if they are separated remotely in their environment habitat, then geographically, then they are separated. So like in the case of frogs, their habitats are different, so they will never, and then they develop into two new distinct forms. So nowadays they established their independent separate species like that. And this figure, which I originally have shown, there's two different flies. One is hawthorn fly, which only feeds and mates only on this particular plant, hawthorn fruits plant. And on the other hand, there's blueberries, another fly only lives on a blueberry, so they don't have any incidents to meet each other. So then over time now they will establish certain specific independent species. Actually that's the cases, and even in these days lions and tigers are two independent species, but physically they can interbreed and produce. In most of occasions they can even produce a viable and fertile offspring, but it was simply just a matter of this, the separation, geographical separation over long history, over their living history. So that's how they became a two independent separate species. It's just that you can like anticipate. And in the case of a temporal isolation, it's a timing, as you can see. So like some are nocturnal and some others are diurnal, so they usually just live in the days, and others, even though they are very similarly related, but one form is only living in the night, like their activities mainly happening in the night, and the others are on the other hand in the days, and they don't have any opportunities to have this event held. So like two skunk, the seasonal differences also, like this particular form of skunk mate only in late summer, whereas this another form of, even though they live in the same area, but this another type of skunk, which is different species, as you can see in this scientific name. This is a gracilis, and another one is a Putorius, indicating two different species. But these guys mate only in late winter, so then yeah, they will never mix their gametes together because of this reason. And behavior also prevents a lot of the bird in particular. They have this very sophisticated and specific ritual, the courtship ritual like this. And so if you do not follow this specific protocol of their own specific unique courtship ritual, then you are not going to be allowed to have this mating chance. Um, so maybe this kind of a very interesting behavior was a result of a long-term evolution, so that in this way they could increase their chance of success in the mating, like not wasting their really pricey the gamete, which to which they have actually invested so much energy. But anyway, as a consequence, you have to have developed a very very unique, specific way of this ritual. Now, those three were as I said, that's something that acting as a barrier by simply not allowing any even the opportunity of this mating chance. But this, even though this is a pre-zygotic barrier, these two cases are something that still they do have this opportunity, but even after this opportunity, something physically it is incompatible in this mechanical isolation barrier, and chemically, in the case of a gametic isolation barrier, chemically incompatible. So in the example of a mechanical isolation, there's some morphological differences prevent successful mating, whereas so like this snail, two different snails, notice that the spiral orientation is different, these two, even though they are highly closely related to snails, but right-handed and left-handed way of spirals indicate that there are certain differences in physically. So their genital opening doesn't match, doesn't align, so they cannot successfully have this mating event. Physically impossible. In the case of a gametic isolation, sperm and eggs can meet, but physically or biochemically they can. For example, like in the case of this sea urchin, one species of the sea urchin sperm cannot bind solidly on the surface of the egg of the other, so they will fail in the fusion of this, like that. So you can imagine many such a similar event of sperm and egg, even though they can actually come closely together, but specifically chemically incompatible, so they cannot make this fusion thing get going, is the example. Now in the post-zygotic barriers, three different occasions, although they are kind of similar. So one is they can have this hybrid, yeah, so they can mate, so they can produce this hybrid between two different species. But the problem is this offspring, the baby, has a viability problem. So they will this offspring is not going to be able to live even after birth. So this viability problem, then you will never be able to produce any offspring. So according to the rule of this biological species definition, you are not one of these same species. On the other hand, here, physically and health-wise is okay, but it is a sterile. The offspring is not able to produce any its own offspring. Then you cannot continue on this gene passage onto the next generation. So there is no meaning of this any, like, you have to be able to pass your gene on to the next generation, as a, like, if you were to be a the legitimate member of this any the organism, especially not to mention any member of a legitimate species. And now this hybrid breakdown is something a little more a little bit interesting case, in that initially they can have a success in this mating, but then next generation, all of a sudden, problem occurs. Is what you see this in the case of hybrid breakdown. So these three events. So in the case of hybrid viability, so the offspring cannot follow a proper development. So this example is one the this salamander, a particular salamander of two species under the same genus, have such problems, so that whenever you have this attempt of a mating, the resulting offspring is just cannot be born, or even it is it can be born, but the health is so challenged, so which is the frail, so unhealthy, so it just it will soon die is the problem, so it cannot go through. Here is another example of this tiger, between tiger and a leopard. Occasionally, naturally in nature they will never mate to each other. However, you can force them to mate in captivity, like in zoo. So a very rare report have been made. Like the most famous report instance was something in 1950s in Japan, in a zoo. It's the actual photograph of this hybrid between tiger and leopard. It's called a leopard, but it has such a viability problem, so it cannot really has a long life. It will probably soon die. Is the such another example of such a viability problem preventing the interbreeding. And here, reduced hybrid fertility is sterility is the problem, even though the health is not a problem, and is it's very famous example we can see in the case of the mating between horse and donkey. Actually, two different occasions. And the male horse, no, I mean male donkey, is called the jack, and the female horse is a more familiar occasion of this mule, which is like an intermediate form between a horse and donkey, and it is really sturdy. So it's been produced at so many occasions, but the problem is the mule cannot produce its own offspring, cannot mate, so it's just a sterile, the dead end is the case. The other way around, the case of a stallion is a male horse, and donkey, I like this name, Jenny, female donkey. When these two are forced to mate, then another hybrid, slightly different in appearance, and its name is a henny. So the mule versus the henny, but both, whether it's a mule or a henny, they are all sterile. So they cannot produce any offspring. So that's why these two horse and donkeys cannot be considered as the same species. Actually, there is a reason, specific reason why they cannot produce this sterile, I mean the fertile offspring is because of the number of differences in chromosome. So, like in horse, there are the chromosome number of a horse is 60 something 4 versus I guess the total number of donkeys is 62 I guess. So this is slightly different number of chromosome. So once they have this hybrid, then they have this matching problem. Probably you could understand a little better when we get to the actual cell division, the mechanism of cell division. But here, it's just suffice to say that their incompatibility, the differences in chromosome number, actually is the reason why I have this the sterility problem occurs in the hybrid. How do actually you may actually think mule and hinnies are always it's very easy to distinguish, but sometimes that's not true. So here in this photograph, it is a little bit tricky whether it is a mule or hinny. How do you find out then whether it's just maybe definitely there must be some specific occasions where you need to know how this particular hybrid was generated, whether like the parental origin. And here, the mitochondrial DNA can be utilized. Mitochondria, if you knew already or not, this is the figure cartoon of mitochondria. Mitochondria is like so here in cell you have many different mitochondria and you just zoom in one single mitochondria, then you get to see all several donut-like circular thing, each of which is actually DNA, a circular form of DNA, unlike the chromosomal DNA in nucleus. Usually you can depict chromosomal DNA in the nucleus like this, right? But in your mitochondria, surprisingly, the mitochondria as an organelle, they have their own independent, their own DNA, the genetic material. That's their own, that's theirs. But also the shape, physically their DNA is circular. We will get to that, why these DNAs are circular in shape rather than this linear form of the more typical eukaryotic DNA. We will actually get to that later on. But for now, the point is why and how mitochondrial DNA can be used to distinguish whether it is a henny or a mule. Because this mitochondrial DNA is only inherited maternal. Maternal origin. So whether you are male or female, your individual, your mitochondrial DNAs are all you inherited your mitochondria from your mother. Okay, me myself, even though I'm a male, but all my mitochondria in my old cells, well, I got those from my mom. Not a single mitochondria my dad gave me. Poor my dad. So in this way, if you examine the DNA sequence, then you can identify whether this DNA belongs to donkey or horse. So in this way, you can distinguish whether this is mule or hinny is the thing. So here the point that I'd like to make is all these mitochondria in higher animals maternally only inherited. Okay. And then lastly, this hybrid breakdown is something that, like I said, uh, the first generation hybrids are okay, viable and fertile. But then all of a sudden, the next generation, when this first fertile hybrid generation, they were actually like mated each other or And then all of a sudden, the next generation, the offspring, is like highly has a viability problem as well as this sterility problem. And this hybrid, so-called hybrid breakdown, is usually often is seen in plants, especially in those crop plants like in rice, what is, for example, rice and wheat and all those the crop plants. Uh, the problem, they do have some the opposite problem of called hybrid vigor. Sometimes if they successfully produce the hybrid between two different races, two different types of varieties of a plant like a rice, then the hybrid is like a stronger, healthier, bigger, like this. This generally we recognize as the characteristic of a hybrid bigger. But on the other hand, the other end of this problem, in mostly two different form of these rices actually cannot produce or this hybrid. And also, even if you were successful in producing this hybrid, here in this uh, photograph, these two P's are actually such hybrid, and they are bigger and stronger, showing this hybrid vigor. But then all of a sudden, when these two hybrid were allowed to were crossed, in the case of a plant, we use these terms crossing instead of mating. When they were crossed, this is the offspring. It is really tiny and unhealthy. This is such an example of such a hybrid breakdown. The second generation, why all of a sudden you have this? And scientists don't really know the exact reason, but simply just have a kind of a speculation on uh, like offering this explanation as, oh, maybe such bad mutation have kind of got to accumulate it on the next generation, the F2 here, the second generation. So then now it's begin to show up, and then like being affected by this such bad influences is what their version of experience explanations. Okay. We have seen several such occasions of the uh, these mating barriers, the zygote formation, the cross interbreeding barriers. And then here, by simply asking, can speciation take place without geographical separation? Sounds like it's very dumb questions, because among those several mechanisms that we have seen as this barrier, mating barriers, some of them you can obviously understand that they can happen without even having any geographical separation. Even though, although, yeah, I admit that in reality, the most of those such separation, the like the reproductive barrier, most of those reproductive barrier or reproductive separation is achieved through this geographical, yeah, it is actually geographical separation. That is true. However, can anyone specifically ask that, yeah, can it be happen without any geographical separation? Then, though we have seen so many, at least a theoretical possibility of, for example, like even those who are living in the same local area, like tiny differences, seasonal differences, and mechanical and chemical differences, will all can contribute to this the separation, the reproductive separation. Yeah, so clear answer is a yes. But here, uh, yes, it can happen. But the reason why I'm bringing up this is simply to introduce two different, maybe unnecessary terminology. One is called the allopatric speciation versus a sympatric speciation, because these two may actually show more frequently than not. So allo means different, other. So allopatric means other country speciation. So you understand, whenever it's the allopatric speciation is referred, that you know it is a something that's achieved through geographical separation. Yes, it is. On the other hand, the sympatric speciation means sim means the same. So same country speciation. So that is something that occurring without any geographical separation. Yeah, sure. So here the sympatric, among many different ways of having this sympatric speciation, especially this polyploidy is one of the major factor for having this sympatric speciation separation in the same location. So obviously we have to understand what the polyploidy is. So by definition, polyploidy is the condition having more than two full sets of chromosomes. Because why this two full set of chromosome is so kind of acting as a standard is because everybody, as an animal, usually everybody gets to have, and your normal body cells, you have two full sets of chromosomes, because one set came from your mother and another set when you were like at the stage of this zygote. Zygote was the result of sperm and egg fusion. Okay, so then one set of those sperm's full genetic makeup set came from a sperm, and the other one set came from an egg fused together, so have a one extra set. So it's a two full set. And as I already went over in the first lecture, it is called this, having two full sets of chromosome is called what? Diploid. Right. So we are all diploid, and most of the other organisms are diploid too. On the other hand, gametes. Gametes should shed off one set of chromosome. Otherwise, when they combined together to form a zygote later on, it will be a disaster. So one such an important thing to become a gamete is you have to come up with some very clever mechanism of dumping one set of your chromosomes to become the half set. It's not a half set. Having only one set of chromosome is called the gamete, is a haploid. The term haploid is kind of a sound like indicating half set, but in reality, it is not a half set, but it's a one full set, as opposed to two full set of other all normal body sets have in their chromosome. So this haploid versus diploid, but polyploidy is more than diploidy is what this polyploidy means. So here, usually this diploid is expressed as a 2n here. The n indicate this haploid number of each unique organism. So if this case was in our human, everybody, the haploid number of chromosome is, in the case of human, 23. Okay. So every normal body cell, since they are all diploid, which means 23 times 2, we have 46 chromosomes. Okay, that's how we use this n as a. So whenever you see this kind of a 2n, you know it's diploid, right? So this is one particular plant case, which is originally a diploid organism. Here in this, for the sake of convenience, probably you we have set up this plant cell has only 3 chromosomes, whatever. If the number of chromosomes as originally is only 3, then it is, yeah. So the number of chromosomes, you have 1, 2, 3, 4, 5, 6. You have 6 chromosomes, right? However, among these 6 chromosomes, 2 is identical, another 2 is identical, another 2 is identical, so that the chromosome set wise you have only 3. Okay. So these 2 diploid, usually when you try to produce the gamete, egg or sperm gamete, like I said, you have to dump one extra set of chromosomes, so that you have to become, in the gamete, you have to become a haploid. However, in this case, you have an error. You have an error in producing the gametes. So what's meiosis? It's a cell division, specific mode of a cell division producing the gamete, sex cells. Whenever you want to produce your gamete, you have to go through this specific type of, specific mode of cell division we call meiosis. Opposite. On the contrary, if you are trying to produce a normal body cells, you have to go through another type of cell division called mitosis. We are going to take a look at mitosis. Yeah, the difference between mitosis and meiosis in a little bit more detail in the following chapters, but anyway here. So the important point is, to produce an egg and sperm, you have to go through the meiosis, through which you have to get rid of this diploid status to become haploid. You should have been the haploid, but you failed to become a haploid. The error always happens, mistake happens always. So that now this sex cells became diploid, and then when these 2 sex cells fuse together form zygote, now it becomes what? Guess what? A tetraploid. Then this tetraploid plant, ever since afterwards, is not able to mate with a normal other individual plant of the same species. Now it established whole new its own independent species, being a different, becoming a different ploidy, is a very common mechanism of generating a new species like that. So now you have this diploid and a tetraploid the plant. And what did I say about this meiosis? Through meiosis, when you try to produce a gamete, then this gamete has to be a haploid. But now, in this tetraploid individual, its gamete is diploid. So this one cannot, is incompatible, non-viable, infertile triploid plant will be generated. Actually, you will see this particular individual a minute later, shortly after, when what happens actually if you intentionally generate this one as a tetraploid and then try to do this and produce this triploid, which is sterile. So this polyploidy, which is popularly manipulated in plants, more specifically, because plants are for some reason more tolerant to polyploidy. In animal, a lot of those negative effects occur once they become a polyploid. So usually you don't really get to see any polyploid higher animals, especially in mammals. You don't really see. There's only one occasion of a mammal being polyploidy, tetraploid. But in plants, there are so many different polyploidy has been produced. And what's like beneficiary to the growers, the farmers, is this polyploid individual could become bigger. Yeah, this many flowers, bigger flower, bigger fruit. So that's what actually these farmers want to have. Examples like this is uh, diploid strawberry, but these days strawberries all you eat is none of them are actually diploid, the normal diploid strawberry, but minimum is a tetraploid, which is, as you can see, is much bigger than. But these days, commercially produced strawberries are mostly even hexaploid, not even tetraploid. Hexa means six. So, and even here, look at this. The watermelon, diploid watermelon and tetraploid watermelons, like bigger, higher ploidy fruits are, look at the seeds, much larger, sturdy, big. In the case of kiwi too, yeah, you get to see the original diploid kiwis, but in these days, most of the common commercially produced kiwis are mostly hexaploidy, something like bigger and you know. And so even strawberries, not even hexaploid and octoploid, having 8 chromosomes. I was wrong. And the seedless triploid, you intentionally produce the triploid using the trick that I have just shown in the previous slide. So like the bananas and the watermelon, seedless watermelons are all those triploid intentionally artificially generated to produce these seedless varieties like that. But on the other hand, like I said, in animals, especially in mammals, polyploidy is extremely rare. So in only one reported case is in South America. This is one rodent, like it's very related with mouse, but it's more related with actually hamster. It's one among this genera, one of the species has been identified to have this tetraploid, but that's the only occasion. Okay. Now, about this speciation, finally, let's consider this kind of a usual speed of a species. Does it usually occur very slowly over a long-term period with a slow but surely changes? That's what kind of what this more common sense like tells us as the case, or in some occasion it would be something the other way around, something that can happen very quickly. So these are two alternative scenarios of such tempo of speciation. One is called gradualism, and another one is called, the other opposite scenario is called punctuated equilibrium. Okay, so here in the occasion of a gradual model, species from common ancestor diverge slowly over long period of time, like slow and steady, okay, over time. So we would actually guess that this should be the correct model. In speaking of considering this the mode of evolution from single common ancestor, you just have a very slow change as a result of the accumulation of a mutation then over a long period of time. Yeah, and that is actually true in probably most of the cases. That might be the correct mode of this speciation in terms of this tempo. However, one important thing is that should not be the only way of this so-called the mode of speciation. Another alternative way, version we can recognize as a punctuated and equilibrium model. Uh, what's happening is new species like diverge from a common ancestor very abruptly, quickly. Yeah, it can happen. Like mutation occur, some very critical mutation occur, and then that mutation somehow allow this the multiplication of this offspring so rapidly, eliminating others, and then you can quickly over a very short period of time you can establish one very distinct species. But then once you have established that species, then you don't really have to have so many other miscellaneous little changes so that it can be stabilized over long period of time is what this version of the model predict. And some cases of the speciation follows this particular model. Okay. So we can, in this diagram wise, we can distinguish this gradualism, like from starting from common ancestor slowly, so that ultimately you have two distinct species. But according to this punctuated equilibrium model, you have suddenly, like accidentally, you have a new species build up from within a population of this existing the original population, and then this one can have this independent over time. You don't really have to have the show the any particular noticeable change over time, and the same thing also happens. So later on, eventually, you have three different distinct new species, and we can easily anticipate that both the cases equally possible under the different circumstances. Yeah, that's the conclusion. Both gradualism and punctuated equilibrium can become a, a specific mode of evolution, in particular, specific mode of speciation under different circumstances. Is the message? Okay. And here, uh, up until now, we've looked at the speciation, and which is obviously have the result of natural selection, right? So this is well according to the scheme of that the model of original Darwin's evolution by natural selection. However, human also can mimic such mother nature in like so instead of mother nature, humans can they take the role of this selection force and then perform this so-called artificial selection. Okay, so it is a man-made reproductive isolation. So whatever, if you want to generate new species, this reproductive isolation is a key. Whether this reproductive isolation is achieved naturally, as we have seen so many according to the many several different mechanisms, or you artificially provide this reproductive isolation. In other words, you are choosing which one to breed. It is called the selective breeding. So it's a man-made reproductive isolation by means of a selective breeding is what this artificial selection is all about. Why do you do that? You are interested in developing your ego, what you need, as you desire. So, desired trait. Usually, you are like you are motivated by monetary, like incentive. You want to make more money by producing bigger, healthier crop, more valuable, economically more valuable livestock, super pig, super hens, like that. So you do that to develop the desired trait. So that's the consequence of so many different breeds of dogs to create your like to suit to fit your own ego. That's how so many different breeds of dogs created by selective breedings of from starting from actually this original the ancestor from all these gray wolves. Yeah. Now the point is, yeah, what am I trying to make here in this figure? What kind of point? So both wolf and the domesticated dogs have the same scientific name of Canis lupus. Here, the Canis lupus. What is this? It's a subspecies name. You sometimes to distinguish further in more details about among different things. So yeah, recognize that all these dogs belong to the same species as this Canis lupus, the wolf. Okay, that means according to the biological species concept, they can interbreed. Yes, that is possible. They can interbreed, right? Still, if the right timing and right opportunities were given, right environment. However, this familiar risk means okay, these dogs are something different. We just created artificially to reflect such feature. We just put additional epithet. Okay, there's a subspecies name, Canis lupus. Anyway, the point I'm trying to make over here is yeah, this is the result of our artificial selection. So it is perfectly an example of what microevolution. Right? What did I say about the microevolution? We are not there yet. Through microevolution, you only get to produce some more varieties within species, within a particular population. But you cannot create a new species. However, consider this. Do you really think these two different dog breed can interbreed? But then, according to the biological species concept, aren't this actually? Shouldn't these two considered to be two separate species? Yeah. Uh, Actually, at the time, even at the time of Darwin was well aware of this domesticated pigeon. Original wild pigeons compared with it, you have so many different forms of each of which actually even could be regarded as an independent new species. It's all the result of this artificial selection. Even more striking example is in the case of this vegetable development. All this now, each of these are even the single independent species. Yeah. But they were all originally derived from cultivation of one wild weed. Actually, this wild weed is very valuable to this plant geneticist, the plant molecular biologist like me, because this is one of the fundamental, the first, the prime model plant in the plant kingdom. But actually, all these were like some of them were. So if you want to produce kohlrabi, you just keep selecting the stem, the feature, some of the trait, and then you finally ended up in producing kohlrabi. On the other hand, if you were just kind of more focusing on the flowers, and then eventually you were able to produce broccoli and so forth like that. And in the case of this story of this corn, it's a very interesting story because original the wild type corn was the wild grown plant named teosinte, grown in these days. Actually, you can see it in the field of Mexico, but obviously originally the Native Americans must have actually cultivated this teosinte to produce some form, intermediate form of at least this corn, okay, through this artificial selection. Later on, in the, like I guess it's the 1970s or something, one renowned Nobel laureate biochemist, after his retirement, he kind of showed his personal interest in the origin of this modern day corn. And then somehow he was kind of suspicious of this. So by just simply looking at these two plants, don't really even have any kind of relationship, resemblance. How could anyone think of this teosinte plant like this, looking like this? Was that actually original ancestral form of this modern day corn plant? But then he demonstrated that these guys were actually these two different forms of related species by crossing these two, and gradually over generation he was able to generate all those intermediate form of this corn and the original teosinte. That is actually it was very lucky case. The reason why he was able to succeed like this, the generating all this intermediate version of this corn, modern day corn and teosinte between these, was because still the genetic diversity, even if you over the long, relatively speaking, long generations, many generations of a long period of time of this selective breeding, virtually the gene pool, you have not lost any actually trait, the genes carrying a specific trait, so that you are able to back cross and then make them reappear in this individual intermediate forms. Okay. If somehow through the course of this selection, if some of the key genes were lost, okay, because you do not know which gene is responsible for making any which trait, so that to the greedy eyes of these growers, you only focus on what you want to have. So it is really easy to lose any specific gene. So you're to simply sum up, you are shredding off any genetic diversity. Like you become the final population of all those artificially selected grown cropland. The genetic diversity is sometimes, a lot of times, actually, you don't have any genetic diversity. Then you become uh, you are in trouble. So first off, if you are interested in developing some of the particular traits of your interest through the practice of this artificial selection, as the presence of this genetic variation in the original population is extremely important. Okay, why? Because no matter how long, how hard you just put your effort, if the original starting population, if they don't have any enough variety of gene pool in their gene pool, the specific genetic diversity, because any given trait is only will emerge as a result of several proper combination of different sets of genes. Trait usually is a result of a multiple genes combination. So only when the right combination of the genes were met as a consequence of this selective breeding, then can you see your trait of interest emerge in the form of your outside appearance, and that you can recognize as a trait. But if you don't have anything to play around this shuffling to begin with. If every individual in the starting population was to have like the same combination of the genes, then you will never be able to have this right combination of this gene, so you will never be able to have this successful trait developed. That's the reason. And conversely, actually, after losing those genetic diversity, you have such a disaster having on your precious business can be seen in most recently in the example of these bananas, right? So these modern day bananas, originally in before 1960 something, like 1965 I guess, is there was the one variety, only single variety of a banana named Gros Michel was the only strain or variety of banana. Everybody grew this Gros Michel, okay? And unfortunately that Gros Michel in itself, they have lost all the genetic diversity. This is such a monotonous gene pool. Then what happened was fungal disease, Panama disease in 1965, particular fungal disease called the Panama disease, stroke. Okay, all those banana plantation, devastating, banana plantation like this. Okay, so you're not able to actually rescue any banana because banana seeds are already, bananas are sterile, so you cannot really plant any seed. The only way you can grow the banana is by just simple duplication of the individual the plant. And so later you will only, there is no other choice but you just have another variety of the Cavendish, which is more resistant to this original Panama disease. Okay. Up until now, the problem is new form of this, the different version of this Panama disease, just like the variation of this coronavirus, the fungal pathogen variant came up eventually. It's called the TP4. Now this TP4 is highly effective in attacking Cavendish variety. So all these Cavendish is now endangered. So like in the last 10 years, that's what I've been hearing. Probably, yeah. So with the advent of more sophisticated science and or the better awareness of these farmers, there must have been done some measures must have been done to actually to rescue all these things out of this another second wave of this disaster. So let's hear what happened.
Here at SciShow HQ, we have a little food area for the employees. Sometimes there's donuts, sometimes there's nuts, sometimes some dried mango. But the one thing that never sticks around and is gone as fast as we can buy it is the wonderful, beautiful, noble banana. Unfortunately for us, they may not be around forever. First, the good. Bananas are healthy, packed with nutrition and energy. They fit in your hand and give nice little cues when they're perfectly ripe and are easy to peel and eat. Shocking statistic, the banana is Walmart's #1 selling item, not the potato chip, not Coca-Cola, not 50 Shades of Grey, bananas. They appear to be so perfect for human consumption that Kirk Cameron attempted to use them to prove the existence of God. Of course, this banana was not created by God, or really even nature. Bananas, at least the ones that you see at the store, were created by people. Don't get me wrong, there are wild banana plants, lots of them. They're native to South and Southeast Asia, and there are dozens of species and thousands of varieties. They're just not the ones we eat. Some of those species, as you might suspect, have seeds, because that's what fruits are. They're fleshy bodies containing seeds. So you might wonder, why have you never eaten a banana seed? Well, you have, kinda. In cultivated bananas, the seeds have pretty much stopped existing. If you look closely, you can see tiny black specks. Those are all that's left, and they're not fertile seeds. If you plant them, nothing grows. Today's bananas are sterile mutants. I'm not trying to be mean, that's just the truth. Unless you were alive in the 1960s, hats off to all those older SciShow viewers out there, every banana you have ever eaten was pretty much genetically identical. This is a Cavendish, the virtually seedless variety that we all eat today, but it wasn't always our banana of choice. Until the 1960s, everyone was eating the same banana, it was just a different banana, the Gros Michel, a bigger, sweeter fruit with thicker skin. You might notice that banana-flavored things don't really taste like bananas. Well, they do. They taste like the Gros Michel. The genetic monotony of the Gros Michel crop was its undoing. A fungicide-resistant pathogen called Panama disease began infecting the Gros Michel crop. By the time growers understood how vulnerable their crops were, the Gros Michel variety was all but extinct. The entire banana industry had to be retooled for the Cavendish. Since they're seedless, the only way to reproduce them is to transplant part of the plant stem. And for the last 50 years, we've been good with the Cavendish, because it's more resistant to the Panama disease. However, somewhat terrifyingly, a strain of Panama disease that affects the Cavendish strain that we all eat has been identified. A global monoculture of genetically identical individuals is a beautiful sight to a pathogen. The fungus only has to figure out how to infect and destroy a single individual, and suddenly there's no diversity to stop it or even slow it down. That's led to a lot of scientists worrying about or even predicting the outright demise of the Cavendish. This wonderful, most popular of fruits might completely cease existence. The good news is, we now have a much better understanding of genetics, epidemics, fungi, and pathology. Scientists and growers have already taken steps to protect the Cavendish. Some growers are creating genetically different bananas that might replace the Cavendish crop if it fails, while scientists are attempting to genetically engineer Cavendish plants with immunity to Panama disease. Plus, we learned a lot from the Gros Michel debacle. Infected fields are quickly being destroyed, and new crops are grown from pathogen-free, lab-grown plant stock. So thanks to the people who work tirelessly to grow and harvest bananas and bring them to us so that we can offer them inexpensively to our employees, and thanks to the growers and scientists working tirelessly to make sure that they don't go the way of the Gros Michel. Thanks for watching this episode of SciShow. If you have any questions or comments or suggestions for us, you can find us on Facebook, Twitter, or in the comments below. And if you want to continue getting smart with us here at SciShow, you can go to youtube.com/SciShow and subscribe.
Okay, so here's the final point that as far as this genetic diversity goes, that I like to make in comparison, like in analogy. When we don't really usually say like, oh, there are so much to eat and happily, by just simply in the case of having the quantity of food, some big quantity of food, that is not usually the case. Yeah, when we say, oh, there are so much to eat, so many to eat, we usually have this type of scenario, diversity. Okay, so how important to maintain the genetic diversity in many different ways is the most important thing, the lesson we should actually bear out of the chapter's lecture. Yeah. So actually, human as a whole, kind of mankind, anticipate at the governmental organization level or even beyond. So there have been different efforts to, in an anticipation and in preparation for such event to ever occur, like any kind of apocalyptic disaster, whether it's a natural, like a huge climatic change or some asteroid hit again, or even man-made such apocalypse like a nuclear war. So in the event, so that at least we preserve some genetic diversity in the form of this crop seed, so that those who somehow survive, who could survive the aftermath of such a catastrophe, could start all over again from scratch is the purpose. So, like for example, I think it's in Finland. I keep just forgetting about whether it's in Norway or Finland. So Svalbard Global Seed Vault was the one of the major such an effort. The worldwide world consortium was made, and then collaboratory worldwide efforts established this, and then collecting all those different crop seed and the local. All these are kind of viewed as a so-called Noah's Ark project. So you know what it means, Noah's Ark project. So this is the outside appearance of such the seed vault in seed storage place in this Finland, this Svalbard. The good thing, advantage of this location is it's such a cold climate, so you don't really need any extra kind of refrigeration, I guess, and freezing. So they have such a goal of collecting such how many different seed samples they are intended to have there in collection. So that was the data of 2017. So by now, their like repertoire must have been also hugely increased. Okay? And also locally, for example, like in Russia, in Siberia, for similar reason, they have also their own Noah's Ark project to have this collection of all different kind of seeds. What about our own beloved country, South Korea? We do also have such efforts and organization, so that we, even though we don't have any such cold climate in our area, but we have this mountaintop. So I believe it's in somewhere in Bonghua, the mountain. We do have a similar such a seed vault project is going on. Okay. So next thing is like how to build this phylogeny, especially like using some of those fossil record tools is these things. But unfortunately, it's been already also like over an hour, so I will have to stop over here. And like I said, I really would like to continue on to finish this chapter 3 this week. So I, with my all apology to breaking my promise, so please allow me to have one more session of the lecture after this, so that we can finish the whole entire thing. We can start new chapter next week. How about that?